All right, so this is the second part of the chapter 22 lecture. Remember, this is what the quiz, you will have a quiz on this part that will be on Moodle. There'll be no calculations for Hardy-Weinberg or anything like that, okay? So the first part talks about natural selection, right, and different types of natural selection. So we all know that natural selection is basically what's going to drive changes in alleles and changes in genotypes. So let's talk a little bit more about it. So I just said that, right? It's the chief mechanism for transforming populations, right? So again, there is some sort of genetic change that occurs, right? A mutation, and then the environment or whatever will select for or against that genetic change. So we just have to always remember that it really is natural selection that is choosing which traits are beneficial or which traits give an advantage and which traits basically give a disadvantage. So I think we know all of this stuff, right? So it says individuals exhibit variations in phenotype. We know now that those variations or the phenotypic variations are caused by genotype changes, right? And that genetic changes can then be passed on to other generations. Some phenotypes will be more successful at survival, and they will therefore reproduce at higher rates. And again, remember, we're mentioning phenotypes here because we're talking about Darwin and Wallace, and they didn't know anything about genotype, right? They just knew about what they observed in those different phenotypes. Okay, the bottom line is phenotypes that have an advantage will reproduce at higher rates. Okay. So as a consequence of natural selection, we will have changes in populations. We just talk about, Hardy-Weinberg as being a tool in which population geneticists will use to measure allele frequencies and genotype frequencies in a population. And then they could look to see how those frequencies change over time. So again, I think we know this. So phenotypes that will improve ability to survive will become more common, right? And phenotypes that basically provide a disadvantage, you will basically start to see them disappear and or be reduced, right? And they could actually disappear totally at some point, right? So it says populations may no longer be able to interbreed, and then the result may be the appearance of new species. You're talking about if you see sort of these separate phenotypes that sort of arise in a given population over time, those two separate phenotypes may actually become new species, right? So we talked about the difference between microevolution and macroevolution. So microevolution changes within a population, on a smaller scale in terms of changes in genotype frequencies and allele frequencies. But when you talk about macroevolution, remember now you're talking about the development of new species. Okay, so if, again, this is talking all about sort of natural selection here. And I just kind of said this, right? So if you see that, you can have allele frequencies change from one generation to the next, right? And obviously what's the force behind this is natural selection, right? So this will shift these allele frequencies. Okay, I think this is a little redundant, sorry. All right, I think I already kind of mentioned these things ahead of the slides, right? So again, you can use Hardy-Weinberg to kind of assess some of this, right? So if you talk about fitness or an individual's genetic contribution to future generations, you can sort of assess this using Hardy-Weinberg, right? So if you look at a homozygous recessive individual who will die before producing offspring, right? So this is a lethal recessive allele. You'll see that recessive allele will decrease in each generation, right? Obviously. So obviously, if this recessive allele is going to cause death, over time, you're going to see that it ultimately is going to go away because there's no way for those people with that homozygous recessive individuals to be able to reproduce. And obviously, being homozygous recessive for this gene provides no advantage. In fact, it puts you at a severe disadvantage. Okay? Now, it doesn't always have to be so severe, right? If you talk about, sort of different degrees of selection. So now this is a recessive allele that obviously, it doesn't provide a advantage, but it's not gonna be lethal. Okay, so you can kind of see sort of strong versus weak selection, right? So if you look at this curve here, right? You can see, again, this is now looking at, again, using Hardy-Weinberg to assess the frequency of a certain allele, this recessive allele, right, in a given population. And if you look over generations, you can see how that changes, okay? So if you look here, this is saying this is a strong selection essentially against, right, this allele. You can see that it's going to decline rapidly over generations, right? You can have sort of a weaker, and these are weaker and weaker and weaker, right? And so obviously if this having this recessive allele is not going to, you know, offer any disadvantage, you're going to basically see that it's going to persist throughout generations. And again, idea here and sort of connection back to what we talked about in the beginning of the chapter is that you would use Hardy-Weinberg to determine the frequencies of the alleles within these generations, okay? And then you would compare them, and that's how you could construct a graph like this. Okay, now when you talk about natural selection, there are some different types, and we talked about this in Bio 113. In fact, I pulled a few slides, or Bio 111 actually. I pulled a few slides from the general bio textbook, just a few pictures just to illustrate a couple of these. So we can talk about directional selection, stabilizing selection, and disruptive selection, okay? And so if we, it draws your attention to figure 22.9, we'll look at that in a second. So in directional selection, you'll see that phenotypes on one end of the spectrum become selected for or against. So, and this is usually due to changes in the environment. One really good example, and it's the same example that's given in the general bio book, is beak sizes in finches during dry. So if you look at the Galapagos Islands, and you track the sizes of the beaks in those birds during dry years versus wet years, you can see that, you know, you have this sort of phenotype that's selected for in the dry years, right? So if you look at beak depth, right, so the size of the beak essentially, in dry years, you're going to see that that increases, and then it decreases in wet year and increases again in the dry years. And so again, you see this selection for this larger beak size, right? Because obviously it gives them the ability to get to those to their food source a little bit easier. So it's basically showing you that it's one end of this spectrum becomes favored, right? This sort of increase in beak size. When you talk about stabilizing selection now, this is where intermediate types would be favored, right? So both extreme phenotypes would be selected against, and the sort of median or mean kind of intermediate phenotype is what is selected for. The best example of this is human birth weight. So if you look at this figure, this is actually showing you this study where you can see, so I guess it was over about 11 years, but this is showing you percentage of mortality. This is birth weight, right? And this is percent of births in the population. So if you look, there's a lot more infant deaths. Obviously, if you're at a really low birth weight, right? You see that there's hardly any infant deaths when you're in this middle range, but then it'll start to increase as you get, you know, as you see these higher birth weights. So bottom line is this is showing you that this intermediate weight is favored compared to being on the low end of the spectrum versus the high end. And if you look at births, you can see the same trend, right? There's more births in this intermediate weight than there are in the low end or the high end. Okay, so that's what stabilizing selection. The other one, disruptive selection, you'll sort of see that both phenotypic extremes are selected for. So in the first one we talked about, it was just one phenotypic extreme, right? Meaning the large beak size was selected for. In this case, it's both extremes, right? So those intermediate ones are actually selected against, but you see a sort of advantage and an increased selection for both phenotypic extremes. So the genetics books use an example of low and high bristle number in Drosophila, okay? And, oh wait, so let me just look a little bit more about that. Anyway, I'm just looking to see. Yeah. So, I mean, obviously it affects their breeding. I don't know that much about bristles in Drosophila, so I meant to look that up a little bit further, but I gave you guys another example as well. But bottom line is, if you follow the generations here, you can see that both phenotypes, you see numbers of, increase in both the low bristle versus the the large amount of bristles. And so, and then you kind of start to see this decrease in the intermediate size. So you see both extremes are favored over time. There's also an example. This example is actually from your general bio textbook. I think maybe this is a little bit easier to conceptualize in the snail population, okay? And so these guys live kind of in two different, they're sort of two different habitats, right? And so obviously the change in pattern or color of their shell is going to help, you know, hide them from their prey, right? And so kind of depending on what region they're in, you're going to see this favored versus this, but it's the same population. They just happen to be in sort of two different environments, right? So again, you'll see after time that yes, this guy's favored and this guy's favored because in his little environment that he lives in, this is going to be more advantageous. And in this guy, in his little region that he lives in, this is going to be more advantageous. Okay, so I think this is probably I don't know, more easier, an example that's easier to relate to, or easier to understand. Okay, next section is talking more about mutation now, okay? So, again, we know that mutations are what drive natural selection, okay? And so, and if you look at a population and you look from generation to generation, you'll see that the gene pool will sort of be reshuffled, okay? And that mutation can actually create new alleles within a gene pool. So remember, if you're talking about gene pool, you're just talking about what alleles exist there, right? It says most mutations are recessive, okay? So most of the time they will be recessive. And basically we can use, there are methods and calculations and statistics that we can use to estimate mutation rates, right? And so you can imagine, again, this is probably particularly useful when you're talking about genetic diseases, right? You want to know how, you know, what the probability is or what the chance is that, you know, these mutation rates might lead to, you know, an increased frequency in a certain disease. So here it says several conditions for accuracy must be met for, oh, you got a redundancy there, sorry, when measuring the force of changing allele frequencies, okay? So bottom line is the allele must produce some sort of distinctive phenotype, right, that basically will sort of set it apart from other similar phenotypes. It says the trait must be fully expressed or completely penetrant. So bottom line is you know, the mutation has to be identifiable. And it says identical phenotype must never be produced by a non-genetic agent. Bottom line is you have to be able to detect or see that this genetic change is correlating to this disease or to this phenotype, okay? And it can't be mistaken for by some other non-genetic agent. And so given that information, you can calculate these mutation rates, okay? So it says it's the number of new mutant alleles per given number of gametes. So again, if you know what a mutation rate is of a given, you know, allele, you can then see how that's going to change from one generation to the next, okay? And it can sort of act as a predictor. Okay, so the one example, and we're not going to do a calculation or anything like that, but the one example the book gives is a dominant form of dwarfism, okay? And so this, you know, affects it's a skeletal disorder. You have an enlarged skull, but you have short arms and legs. They diagnose it by x-ray at birth, and basically it says fulfills requirements for measuring mutation rates. That's what we just talked about before, that given the way that this is expressed, you're actually able to calculate a mutation rate here. And so when they looked at about 250,000 births, they basically estimated this mutation rate as about 1.4 times 10 to the minus fifth. Okay, and so again, from there, sorry for switching there, but from there, they can then predict how the alleles might change in a given population over time, and how, you know, if you're going to see this form of dwarfism, you know, if it's going to become more frequent, things like that. Okay. All right. Now let's talk about some other things that can alter allele frequencies. So we just talked about mutations. We probably knew that already. But there are other things like migration and gene flow that can alter population or allele frequencies. Okay, so if you look here, I mean, what's migration? I think we know. Basically, you have individuals moving between populations. You know, it's a species divides into populations that are separated geographically, right? So you can have a just geographical separation of populations, and then basically over time, you may see that these allele frequencies will start to change, right? And so there's an example. Book gives an example here of blood type. So it's showing you the B allele of the ABO locus, right? The blood typing locus. You see this highest frequency in Central Asia and lowest in Northeast Spain. And basically those frequencies parallel the sort of the way that these Mongols migrated into Europe. Okay, and so it's sort of this genetic relic of history. So if you look, oh, I thought I wrote that here. Yeah, so wait, you see that, highest frequency in Central Asia, and lowest in Northeast Spain. So what this map is showing you is the frequency, those percentages are the frequency of the B allele, okay? And so you can see that there's much higher frequency over here than there is over here. And again, this is basically due to the way and the pattern of migration of these people. And so you still see this sort of alteration in allele frequency based on that. And so it's illustrating the sort of effect that migration and migratory patterns can have on the frequencies of alleles in a population. Okay, another thing called genetic drift. We talked about this before. And particularly this is going to affect small populations, okay? So you'll just see these random changes in allele frequency due to this. And so this is just possible by chance alone, okay? It says degree of fluctuation will increase, right, as population size decreases. Obviously, if you have a really small population and something happens to wipe out, half of that population, it's going to have a more dramatic effect on the frequencies or the percentages of the alleles that exist, rather than having a larger population. So there are sort of types of genetic drift, right? Founder effect and genetic bottleneck. If you talk about founder effect, this is basically a small number of individuals leave a population, and then they basically start reproducing, that small gene pool starts reproducing from there. So that gene pool may not even be representative of a larger population at all, because it only depends on which people left that original larger population to start the new one. Bottleneck, right? This is some sort of drastic change that occurs, maybe like a natural disaster or something like that. Again, same thing, you're going to sort of wipe out a number of individuals within that population, and then what's left, obviously, is going to be what reproduces, right? And so in some cases, those individuals that are left may be very different genotypically than what was in that original population. So this is one, easy sort of picture, again, pulled from the general bio book, right, where you can see here's your population of frogs here, right? And here's our brown frog. Right now, he's about 10% of the population, right? Natural disaster occurs, and it kills off some frogs. It actually never kills off any brown frogs, that one brown frog here, but it basically kills off some of the green frogs. Now you can see that instead of this brown guy being 10% of the population as before, now he's 20%. And so that's essentially doubling the allele frequency in a short amount of time, okay? So again, the point is, this is a process that changes allele frequency. So due to this natural disaster, the frequency of the alleles has changed dramatically. If you talk about, genetic drift in humans, right, here's an example. So this albinism, a certain specific type of it called OCA, okay? And it's predominant in the Navajo tribe that apparently that lives in the northeast Arizona, okay? Now, it's found that the frequency of OCA, right, so in the Navajo tribe, it's about 1 in 1,500 to 1 in 2,000. In the white population, it's only 1 in 36,000. In African Americans, 1 in 10,000, okay? So obviously you can see it's much higher frequency in the Navajo tribe than in other populations. Now, there are 4 different forms of this OCA, so OCA 1 through 4, all of which sort of have varying degrees of melanin deficiency. The one that we're going to focus on is OCA 2. So this OCA 2 is caused, we know what the genetic mutation is and where that genetic mutation is. So it's caused by a mutation in this key gene, we're calling it. And there was a study done where researchers screened for mutations in this key gene using PCR, okay? So they looked at certain individuals. They surveyed about 134 normally pigmented Navajo and 42 members of the Apache tribe, which was a closely related tribe. And they only found this deletion causing OCA2 in the Navajo population. So they didn't find it in that closely related Apache tribe. And if they looked at other tribes or other people that weren't even a member of that Apache tribe or the Navajo tribe, they basically never saw that deletion. So that from this, they calculated that the heterozygote frequency in the Navajo tribe is about 4.5%. Okay, and so this research indicates that that mutant allele probably came from some founder mutation about 400 or 1,000 years ago, right? There was some, small number of individuals that sort of broke away to form this tribe, and they expressed that mutant allele, and then obviously started to reproduce, and that's why you only see it sort of isolated in that Navajo tribe. So if you look at this picture here, this is a representative gel from their PCR experiment, right? And you can see here N4 is an affected individual, meaning it has that person has that form of albinism, OCA2, right? Did I say it wrong? Yeah. Here, this guy too, N5 is affected, and so you can see that they just have this one band here, and okay, so you can see, so they only have this one band, and they have this deletion, right? So compare it here to the normal control, right? Normal control has this nice thick band here, this 257 base pair band. Okay, so this guy, is a homozygous dominant here, right? No mutant allele. Okay. So again, you can see this mutant allele, or the, recessive allele, I guess, in the N5, right? No normal allele, right? In the two affected. And if you look at the parents of N4, you can see a band here, but then also a band up here, okay? Indicating that they're heterozygotes. And so bottom line is you can use this method to genotype, a lot of the individuals in the population to come up with the data that they came up with here. Okay. Another thing that will change gene pool, right? So not necessarily allele frequency, but will change genotype frequency is non-random mating, right? So this is if there are certain individuals that will only mate with other certain individuals within a given population. Again, you're not going to change allele frequencies, but genotype frequencies will change. And then it says subsequent selection for or against certain genotypes could then affect allele frequencies, but non-random mating itself will just change genotype frequencies. Okay, that's what this is telling you here. And so some forms of non-random mating, positive assortative mating, so similar genotypes are more likely to mate with other similar genotypes rather than dissimilar ones. You also have the other side of it, right? Well, where you will see dissimilar genotypes more likely to mate than similar ones. And then also, obviously, inbreeding, okay? And, if you're talking about inbreeding, particularly, you're going to see an increase in the proportion of homozygotes in a population, right? And this is particularly important in genetic diseases, right? So you will see a much higher prevalence of genetic disorders in a population in which there is inbreeding, okay? Because you're, that gene pool or the variation in that gene pool is limited. And so I could have pulled up an example here, but I didn't. I think we all can understand that, right? So you can look at a pedigree from a population that inbreeds, and you can see that there's a much higher prevalence of, a given genetic disorder if you look. Okay, if we talk about another thing that's going to sort of affect the gene pool, allele frequencies, but then also particularly can lead to the development of new species, is reproductive isolation, right? So, so basically, if you have two separate right? So you have population, right? And then ultimately, part of the population starts to sort of diverge from each other, and you kind of end up with these two separate populations. Over time, they may lead, those two populations may actually become separate species. And so they may then, start to become reproductively isolated from each other, right? And only able to breed with that population. And again, what was a single species before will end up dividing into two separate species. All right, so, I pretty much said this, right? So when we talk about the development of new species, you term that speciation, right? So again, two populations start to become more and more different over time. Then they start to be only reproduce with each other, and then eventually over time they may develop or lead to the development of two new species, right? So this is kind of just showing you this here. Okay, that you can kind of see these this sort of division here of these species here. And so obviously we call that macroevolution. And again, I kind of said that before. Okay, so when you talk about the development of new species, you're now talking about macroevolution. When you talk about changes in allele frequencies and genotype frequencies within a given population, you're talking about microevolution. And like I said, you may see in a population, kind of these two separate groups that start to arise, but until they actually form separate species, it's not macroevolution. Okay, and usually, macroevolution obviously is going to happen on a much larger scale and takes a lot longer than microevolution. Now, there are, various barriers or reproductive isolated mechanisms that exist to basically, separate or divide populations and then species. Okay, so ecological, behavioral, seasonal, mechanical, physiological. We've talked about all of these guys before, right? So if you talk about a prezygotic isolating mechanism, that will prevent mating from taking place at all. Whether it's because they mate at different times during the year, whether it's because they are in different geographical regions and they don't come in contact with one another, they have different mating calls or mating rituals, right? That's a behavioral one. Those are all prezygotic. If you talk about postzygotic, the two populations can mate with each other, but they actually never produce a viable offspring, okay? Or they produce a sterile offspring that can then cannot reproduce from there, okay? An example of a geographical change leading to speciation is this example of snapping shrimp. So if we look at, I think it just shows you the picture there. So prior to the formation of this isthmus in Panama, members were a single species, okay? But when this closed, it basically divided that species into two separate populations. And then over time, you had basically those two populations become separate species, okay? So it says here, sorry for the redundancy here, but it says 3 million years of separation resulted in completely or nearly complete speciation, okay? But look how long it took. It took 3 million years, okay? And again, there was a geographical barrier that separated the two populations, and then over time, they actually developed into new species, okay? So let's show you a little picture of the snapping shrimp there. So, I just told you that example was about 3 million years, right? And if you kind of talk about average time of speciation, it says about 100,000 to 10 million years. However, there are examples in which speciation has occurred on a shorter time scale. So some examples here, and we'll talk a little bit more about the fishes in the East African lakes, and we've given that example in general bio as well. There's examples for marine salmon, palm trees on isolated islands, polypoid plants, and also a brown algae. So there are many, and there's more, right? These aren't all of them, but there are many examples of speciation occurring over a lot shorter time scale than 100,000 to 10 million years. Okay, so here's an example with these fish. So there's a particular lake, Lake Apoyo in Nicaragua, that basically formed within the past 23 years. And it's home to two specific species of fish, okay? These Midas and these arrow cichlids. And so these guys are common in the region in general. These arrow cichlids are only found in this one lake, this Lake Apoyo. So if you look here, right, you can see this lake here. Here's the sort of larger lake. And so this obviously sort of formed later on, right? This sort of small subset, this little lake here, from the larger one. And so they've, scientists have really studied this population and try to figure out, right, what the origin is of these guys that live in this isolated lake, right? Where did they come from? And so they've used phylogenetic, morphological, ecological analysis, and also DNA data, sequence data, some more molecular evidence. And they basically concluded that those guys evolved from the Midas cichlids that were, I guess those guys are found in all the lakes kind of in this area. Okay, but the bottom line is they were trying to figure out where this particular species evolved from. Okay, and after a lot of research, they did find that they were from this Midas cichlid. And that what this research also showed is that the speciation occurred in about 10,000 years, right? Compared to what we talked about before, macroevolution and speciation occurring on the order of about 100,000 to about 10 million years. Okay, so much shorter time scale. And so if you're curious to see what these guys look like, here's the example. And you can see that they even, by appearance, they look pretty different, okay? That this guy was derived from this guy, okay? So obviously, we can use all of this sort of data, right, to determine evolutionary history. So I'm going to give an example here of in terms of determining which, I guess, species was sort of the common ancestor to terrestrial vertebrates, right? So, it's a I guess, highly studied area in terms of, again, if you think about, the origin of life and stuff happening in water, right? And then at what point, where was the branch point from organisms moving from the water to the land, okay? So obviously we can construct phylogenetic trees, and we obviously do that based on phenotypic data, molecular data, all the things we've kind of been talking about. And so from there, you can see these branch points, right? So you can see kind of, right, this is the common ancestor to all these species. You can see these different branch points, right? So at each branch point here, right, or each node here, you can see that this was a common ancestor to species C and species B, right? That both were derived from whatever that, species was here, okay? So we've seen phylogenetic trees before. Hopefully we understand how to interpret them, okay? But you can see that, species A, whatever this branch point was here, right? Species A diverges off, and then these guys that led to species B and C. So it's sort of a separation point here. Again, remember that these phylogenetic trees are constructed based on DNA sequence, based on phenotypic data, based on behavioral data, right? Based on a whole bunch of things, not just DNA sequence, maybe protein sequence as well, okay? And so there are many tools that scientists can use to basically align DNA sequence to see where changes are and then to construct these phylogenetic trees. So as I mentioned, one of the sort of areas that's studied a lot is the transition of vertebrates from the ocean to land, okay? And the question kind of is, well, which fish, lobe-fin fish, crawled ashore basically as the ancestor to all of these vertebrates, okay? And there's sort of been a few, I guess, candidates, and the biggest one are the lungfish versus the coelacanth or coelacanth. And from more and more data that we've obtained, right, including DNA data, we've actually figured out that the vertebrates are more closely related to lungfish, if I could say that, rather than the coelacanth. And so again, if we weren't able to use this, protein data or DNA data, we wouldn't be able to figure this out. So remember that when you talk about evolution and evolutionary history and constructing phylogenetic trees, it's not just based on phenotype and what things look like. It's really a lot, very heavily based on molecular data now. Okay, so here, this guy, this lungfish, is now believed to be the ancestor to terrestrial vertebrates rather than the coelacanth. So again, you can kind of, we use some of this data to construct a phylogenetic tree. like this. Okay. And this is kind of just like a little brief thing here showing something called the molecular clock. So again, using this molecular data that we talked about, we can sort of construct sort of graphs or molecular clocks that show the rate of evolutionary change. So again, it's kind of, if you look over time and then measure what changes there are over time, you can then kind of construct this sort of, put the divergence of certain species, or when that divergence happened, right? Based on the changes that you see over time, okay? So this figure 22.2 is showing the divergence times for humans compared to other vertebrates, okay? And this is really based on fossil records. So if you look here, and then this is showing you based on molecular data, and actually you see that they correspond pretty closely, right? So if you look here, these are the corresponding numbers, right? It's human versus chimpanzee, human versus orangutan, human versus monkey, human versus mouse, human versus cow. And you can see you can see how the more closely related species obviously are a lot closer in terms of the number of differences in nucleotides and amino acids. And then as you get, towards the zebrafish here, you can see there's a lot of differences. But then it also is taking into account time. Okay. And again, you can see that this correlates, the fossil record correlates with molecular data. Okay. One other thing, that obviously these tools are used to study is obviously the origin of, humans or where we were derived from, where the species of Homo sapiens originated from, okay? So it says genetic diversity so obviously DNA sequence is used to infer where Homo sapiens originated. And we see the highest levels of diversity in African populations. Non-African populations had origins about 50,000 years ago. Basically, the data indicate that Homo sapiens originated in Africa, right, from earlier species of the genus Homo. If you talk about the Neanderthals, they lived in Europe and Western Asia about 300,000 years ago. They disappeared about 40,000 years ago. And, it's believed that they coexisted with Homo sapiens for about 30,000 years in several regions, okay? And there have been studies that basically, again, based on molecular data and fossil record, that, have set up these phylogenetic trees and to see kind of when our species, Homo sapiens, diverged from the Neanderthals. And so it says Neanderthals, from that study, Neanderthals weren't direct ancestors of our species, but Neanderthals and members of our species were able to interbreed based on these studies. And that Neanderthals, the genetics of the Neanderthals or what existed in the Neanderthals, you see that those genes contributed to our genome as well. Now, in 2008, there were actually some fossils discovered in a cave in Siberia. And actually, they completely sequenced the genome of these fossils, and they found out that those fossils actually weren't Neanderthal fossils, and they weren't Homo sapiens, but that they were this separate species, and they named them the Denisovans based on where they found them. And it says that if we look at our modern human population, about 4 to 6% of the DNA in the islands of South Pacific are actually derived from the Denisovans. So, probably we're going to discover more things like this as well. And so if we didn't have DNA sequencing, right, or we weren't able to use techniques like that, we probably would have just thought that those were fossils of Neanderthals, right? But indeed, they were different species. So obviously that changes that phylogenetic tree and changes, our ancestry. Okay. All right, so like I said, probably, we may find more species like this, right? There are a lot of, now, once we discovered these, fossils, now there's a lot more studies going on to basically determine, how, the relationship between this new species and other species that we knew about already. And so that concludes it. Like I said, I will post a quiz, and it will be pertaining to everything in this chapter, not including Hardy-Weinberg. No calculations, just all the sort of content that we just talked about. Thanks. That's, oh, sorry, that's showing you the cave where they actually found these fossils.